All right, everybody, welcome to the video here for lesson number 10. So in lesson number 10, we're going to do something called the difference of sample means. We're going to evaluate another statistic of our samples that we can find. So far we know how to find the statistic of finding the proportion of a sample. We also know how to find the statistic of finding a mean of a sample. Now we're going to find the difference of sample means when we have our samples split up into two groups. So the opening exercise says, explain why the following statistical study is an experiment. So I want you to pause the video, read through the scenario, and tell me why it's an experiment. All right, so I'm hoping that you listened and you took the time here and actually read through this, and you're now just checking what you wrote down here against what I'm about to say. So it says that we have these 10 tomatoes varying in shapes and sizes. Some of the tomatoes were given a nutritional experiment where there's an application to yield larger tomatoes, and 10 were just in regular conditions. So the reason why this is an experiment, other than it says the word experiment in there, is we're testing for cause and effect. You could have said we're looking for a causal relationship. You could have said there's a test group and a control group, right? The test group meaning the plants that got the additional supplements and the plants that got just regular water and sunlight. So here are our tomatoes. We have 10 tomatoes. All of their weights are shown, and we're labeling them as numbers 1 through 10. So for right now, we're not concerned which tomatoes receive the additional nutrients. We're just going to randomly assign the 10 tomatoes into two groups, because we want to test and see what the difference in the means are of the two groups so that we can do a computer simulation and try and figure out if there is a significant difference when we put the tomatoes into the two groups, where we only put tomatoes with the nutrients in one group and the tomatoes with the no nutrients in the other group. So we're looking for a statistical effect here. So right now I'm just randomly selecting. So what I did for you already is I used my random number generator on my calculator, and I randomly selected 5 numbers between 1 and 10. And I got numbers 1, 4, 5, 7, and 10. I made those group A, and then the remaining 5 tomatoes become group B. So it says confirm the mean for group A. So if you add up your 5 numbers in group A and divide by 5, you should get 6.76. We call that the sample mean of A. We can also find the sample mean of B. So if you add the 5 numbers up in group B and you divide by 5, check it, you should get 6.48. So our new statistic that we're finding here is what's called the difference of sample means. So we have a mean of A, we have a mean of B. I want to find the difference of them. So to calculate the difference of the sample mean, we are literally just going to subtract the two numbers. Remember, I've been saying this all chapter. The math that's involved here is not hard. It's adding, subtracting, and multiplying by 2. It's what we are getting from this that is telling us something. So the difference of these two samples, the statistic that we're finding here, is 0.28. Okay, so on the next page, I'm explaining a little bit further what we're finding here. So the statistic of interest that we're looking at here is the difference in the two groups of tomatoes. Now, if I reorder them, if I re-randomize what I just did on the previous page, I can get different sample means and subtract them. So depending on what the difference in sample means is, it's going to tell you something different about the two samples that you had. So for example, if I had a difference of sample means of 1.64, that tells me that the sample of A has a larger mean by 1.64. If I subtract the two sample means and I get a negative number, that means sample B is larger. And it's larger by 0.4. If I subtract the two samples and I get 0, that means the sample A and the sample B are the same value. So what we're able to do is we're able to randomize these tomatoes over and over and over again to figure out the difference in sample means. So below we've done it again. So I've used my random number generator. I've randomized so that I can get sample A and sample B as two different groups of tomatoes. Again, still not knowing which ones have the extra nutrient and which ones don't. So what I'm going to ask you to do here is take a minute and try and calculate this statistic. So find the sample average, the sample mean of A, find the sample mean of B, and then calculate the difference of sample means so that we can see what this particular re-randomization of the samples gives us. So take a couple seconds to do that. All right, so if you did this correctly, you should get the sample of A has a mean of 7.84. The sample of B has 5.4. And the difference of sample means is 2.44. Meaning sample A has a mean larger than sample B by 2.44. I hope you're with me so far because we're just finding averages. We're not doing anything crazy. So on the top of the next page, if we were in class together, I'd actually have you use your random number generator on your calculator and re-randomize this a couple more times to get more samples. Luckily for us, we're able to do this on the computer. So I've already computer generated a randomization of these 10 tomatoes a bunch of different times, 250 times to be exact. And these are all dots representing the difference of sample means of those re-randomizations. So re-randomizing is us just taking the 10 tomatoes and putting them into different groups and seeing what our sample is. So looking at our sample distributions here of all of our difference of sample means, we can see that it's an approximate bell curve, it's an approximate normal curve, and the center seems to be right around 0. Right? So we could say the approximate center is 0. Whenever we're doing a difference of sample means, a lot of times the approximate center becomes 0 because when we re-randomize, we're going to wind up getting those groups that are going to have that 0 difference. So what does this all tell us? What are we looking for here, right? So on the next page, it gives us some probability questions. What are the chances of our difference of sample means being 1.64, negative 0.8, or negative 2.4? So if we look for 1.64 or more, so if I look at our randomization, 1.64 is somewhere around here. We can count up how many dots are in there. So this would be more than or equal to 1.64. So luckily for you, I've already counted the dots, and I got 17 dots out of 250. If we do some quick calculating, we could even figure that out as a decimal. So it's about a 6.8% chance. If I look at the dots that are at negative 0.8 or less, so at negative 0.8 or less, so there's a lot of these. Lucky again, I counted them for you. So if we count up all those dots that are less than or at negative 0.8, there are 67 of them, which gives us a chance of that happening of about 28.6%. And lastly, at negative 2.4 or less, so if we look at negative 2.4, there's only 4 dots. That's an easy one to count. So we get 4 out of 250, which is roughly a 0.016% chance. So again, what does all this mean? So when we're looking at these probabilities, when we're looking at our chance of our difference of sample means being within or more than or less than certain values, we're looking for statistical evidence that tells us that there's a significance within the sample means or not. So the standard cutoff for determining statistical significance is 5%. Think 95% confidence interval. We're looking at the numbers that are outside of that confidence interval. Okay? So if we have numbers that are outside of the confidence interval, we're talking about something that would be an unexpected result, meaning there's statistical significance in that group. So when we actually now put our tomatoes into the correct groups, a treatment group and a control group. So the treatment group has the actual nutrients added to it. The control group does not. I can calculate the statistic for the actual tomatoes in their proper groups now. So if I find the average of group A, the average of group B, and then calculate the difference in the sample groups, I can determine whether or not that difference is significant or not. So when I find the statistic here for the sample of A, I get 7.74. So again, add the 5 tomatoes up, divide by 5. In group B, if I add the 5 tomatoes up and divide by 5, I get 5.5. And if I subtract to find the difference of sample means, I get 2.24. That is my difference of sample means. So the interpretation of this difference is saying that the treatment group, on average, is 2.24 ounces heavier than the control group. So the treatment group is bigger in average than the control group. Now what we want to do is we want to calculate and see, is this statistically significant or not? Right? Is this within the 95% confidence interval where we're saying we expect that difference to happen? Or is it outside of the 95% confidence interval where we're saying we don't expect that difference to happen, meaning there was a significant increase in size by adding the nutrients? So what we have to do is we have to calculate a probability. So we have to calculate a probability that the difference of sample means is greater than or equal to 2.24. So what's the chances of us getting this difference or more of a difference? So if I go back to my dots again, there's only 6 dots that are approximately at 2.24 or more out of 250 dots. So we get approximately 0.024, which is about a 2.4% chance of happening. So because there's only a 2.4% chance of this happening, when we re-randomized and put our tomatoes into random groups that didn't have treatment, had treatment all over the place, we see that when we put the groups back into their original groups of a treatment group versus a control group, there's a significant difference here because it's outside of the 95% confidence interval. So what we can say is the treatment group has a significant difference in mean weight compared to the control group because the difference of sample means has less than a 5% chance, that's a 5, not an 8, 5% chance of happening. If I calculated this difference of sample means and it gave us a different value that gave us a percentage or a chance of happening that was more than 5%, we say there's no significance here statistically that shows that the treatment group got something that the control group did not. So essentially we're using statistics to prove that the additional nutrients made it so that those tomatoes grew to a larger size. I'm hoping this makes sense. What I'm going to do for you though is tomorrow in class, you're going to work on the homework assignment for lesson 10, so don't do that tonight. And I have another video going through this, the homework problem so that you can see kind of another problem like this and work through it on your own. Okay? If you have questions on this, I promise I'll answer them in class the next time I'm in on Wednesday, right? But for right now, if you have to, look back at the video, see what I did to calculate this statistic. It's not as hard as it looks, okay? Hopefully this makes sense, and I'll see you all soon.